Throughout the world, one animal has cemented itself to be the ultimate companion for humans. One animal has made itself the most respectable, most adaptable, most interesting animal to coexist with humans: the dog. Today, we will be looking specifically at a group of dogs that align more closely with interesting than respectable. Dogs that are underproportionately built. Dogs that would struggle reaching the top shelf. Forget it. They're just short dogs. Today, we're going to learn why they are half-sized. Our specimen is a Corgi Beagle mix named Huxley. And don't worry, all of these short jokes won't hurt his feelings. They all go over his head.
So why are these dogs bite-sized? Well, before we can learn what went wrong in development, we must learn what should happen. It all starts in the nucleus of the cells. Within the nucleus is their DNA. DNA is the genetic material that codes for all functions and growth of an organism. It is made of a sugar-phosphate backbone structure and is connected by nitrogenous bases. These bases are paired complementary. This means that the structure of one base complements and connects to another base. Adenine and thymine are complementary. Cytosine and guanine are complementary. DNA codes should be expressed like this: TCG, AAT, and CGT.
When duplicating, enzymes are used to produce two strands of DNA. These enzymes all have very specific roles. Helicase unwinds the DNA double helix. Primase lays down RNA primers to initiate synthesis. DNA polymerase synthesizes new strands by adding nucleotides. And ligase seals gaps, which are the Okazaki fragments, in the sugar-phosphate backbone. This DNA is very precious and only stays in the nucleus to ensure it does not get damaged.
You may be wondering, how in the world does the body then get its genetic information? Welcome to the wonderful world of RNA. It is a nucleic acid present in all living things, acting as the messenger between DNA and protein production. It replaces thymine and uracil. It takes the DNA information using complementary pairs, moves through the cell, and then delivers the information. This is called mRNA, or messenger RNA. This process is called transcription. It is like copying a recipe from a cookbook. You get the same information, but you are not using the original copy.
In the world of RNA, there is also tRNA and rRNA. tRNA acts as an adapter, bringing the correct amino acid to the ribosome during protein translation. The rRNA is a component of ribosomes, the protein manufacturing factories in the cytoplasm of the cell.
So during translation, the next step of RNA's use, the mRNA is read according to the genetic code, which relates the DNA sequence to the amino acid sequence in proteins. Each group of the three bases in mRNA codes for one codon, and each codon specifies a particular amino acid. The mRNA sequence is thus used as a template to assemble and order the chain of amino acids that form a protein. So mRNA brings the code, tRNA transfers the code, and rRNA reduces the code. Peptide bonds are covalent chemical links formed between amino acids during translation. The enzyme, yay, more enzymes! Peptidyltransferase catalyzes this reaction, joining the carboxyl group of one amino acid to the other amino acid of the next, releasing water. This process builds peptide chains, aka proteins.
To bring it back to food, if transcription is copying the recipe, then translation would be actually preparing and cooking the food. If all this happens correctly, they should grow to be a normal height. But this doesn't happen. Ooh, I know. It's because they're freaks.
Well, no, but they are mutants, so a mutation is a permanent change in the DNA, where the extra gene that the pup possesses affects the certain patterns in the DNA sequence specifically called introns.
Dachshund Dwarfism is when all short-legged dog breeds have an extra copy of the gene that codes for a growth-promoting protein called fibroblast growth factor 4, FGF4. The extra FGF4 gene of chromosome 18 explains the short-legged phenotype that causes breeds like Corgis, Dachshunds, and Basset Hounds to be much shorter than other dogs.
So our little munchkin here is short because when he was developing, the extra FGF4 retrogene causes growth plates to classify, leaving him in a virtual state of physical puppyhood. Now that makes sense. if you know what the heck a retrogene is.
Retrogenes are DNA genes that were transcribed into RNA and then were mistakenly turned back into DNA. This DNA has the same coding protein sequences as the original gene that produced the mRNA. This retrogene gets put back into the developing genome in a different location than before. This new DNA is capable of producing the FGF4 protein. The overproduction of the protein can cause key growth receptors to turn on at the wrong stage of development. This is what causes the growth plates of growing organisms, or in our case, little short dogs, to harden or calcify early.
In regards to mutation, this is a specific type of mutation. It is not a standard point or frameshift mutation. It is the type called retrogene insertion. Like I explained before, the retrogene is inserted back into the genome, causing extra protein production.
So, with our golfable little buddies, their DNA codes for RNA, which codes for codons, which code for amino acids that form proteins. However, a mutation occurs when the mRNA is turned into a retrogene. This DNA is inserted back into the genome and causes extra growth protein to be made. This excess protein coding causes signals to be sent early in development, causing the Whittle Short Kings.